Welcome to the Science of Ageless Living. I'm Dr. Ethan Hausman-Marquis, Dr. Scientist, and the only peptide clinic physician in London with a published body of work specifically in this field. This is the Ageless Top 20 Peptides series. Every episode covers one compound, the real biochemistry, the honest evidence base, what we know, what we don't, and what the regulatory picture actually looks like. No hype, no discount codes, just the science. Today, GHK-Cu. On paper, this compound should not be as interesting as it is. It is a tripeptide 3 amino acids, glycine-histidine-lysine, molecular weight of 340 Daltons. Structurally, it is about as simple as peptides get. And yet, the biological activity attributed to it in the peer-reviewed literature spans wound healing, collagen remodeling, antioxidant defense, anti-inflammatory signaling, angiogenesis, nerve regeneration, and in a 2010 gene expression analysis, apparent regulation of over 4,000 human genes. That is an extraordinary range of claimed activity for a molecule of this size. Some of it is well supported, some of it is early stage. Some of it requires significant qualification. Sorting those three categories is exactly what this episode is for. Let's do it properly. GHK-Glycine-histidine-lysine is a naturally occurring tripeptide found in human plasma, saliva, and urine. It was first isolated and characterized in the early 1970s by a researcher named Loren Pickart, then working at the University of California, San Francisco. Pickart was studying factors in human plasma that could stimulate liver tissue regeneration in vitro, and he isolated a fraction that had unexpectedly potent regenerative activity. When he characterized it, he found this tiny tripeptide 3 amino acids with a high affinity for copper ions. The copper binding is not incidental. It is central to GHK's biology. GHK binds copper, specifically divalent copper, Cu2+, with high affinity, forming the complex we call GHK-Cu. And it is the copper peptide complex, not the free peptide, that mediates most of the biological activity. Copper is an essential trace element and a cofactor for a range of enzymatic processes, including superoxide dismutase, the primary antioxidant enzyme responsible for neutralizing superoxide radicals, and lysyl oxidase, an enzyme critical for cross-linking collagen and elastin into functional connective tissue matrix. The biological logic of GHK-Cu at its most basic is that it acts as a copper chaperone, a molecule that delivers bioavailable copper to tissues and enzymes that require it, in a form that is non-toxic and targeted rather than free and potentially oxidatively damaging. What makes GHK-Cu relevant to this series is what happens to its circulating levels with age. In young adults roughly 20 to 25 years old, plasma GHK concentrations are approximately 200 nanograms per milliliter. By age 60, that figure has fallen to around 80 nanograms per milliliter. By later decades, it approaches undetectable levels in many individuals. That is not a modest decline. That is a greater than 60% reduction across the adult lifespan. And given what GHK-Cu appears to do in tissue maintenance and repair, which we will come to, the timing of that decline maps onto the timing of age-related deterioration in wound healing capacity, skin integrity, connective tissue quality, and antioxidant defense in a way that is at minimum biologically suggestive. The synthetic version of GHK-Cu is chemically identical to the naturally occurring human complex. It is produced through standard solid-phase peptide synthesis followed by copper chelation. It is not a foreign molecule. It is a sequence that exists in human biology, declining with age, that can be provided exogenously. As with thymosin alpha 1, that framing has implications both for the mechanistic plausibility of supplementation and for the safety expectations. The mechanistic story for GHK-Cu is unusually broad for a molecule of this size, and I want to take you through it carefully, distinguishing what is well replicated from what is preliminary. The first and most thoroughly established mechanism is wound healing and tissue repair. This is the area with the deepest experimental foundation, dating back to Pickart's original work in the 1970s and built out extensively through the 1980s and 1990s. GHK-Cu promotes wound healing through several convergent mechanisms, It stimulates fibroblast proliferation and migration into wound sites. Fibroblasts are the primary cells responsible for producing collagen and extracellular matrix, the structural scaffolding of skin and connective tissue. GHK-Cu acts as a chemoattractant for fibroblasts, drawing them into wound beds and increasing their synthetic activity once there. It promotes collagen and glycosaminoglycan synthesis. Collagen, particularly type 1 and type 3 collagen, is the primary structural protein of skin, tendons, ligaments, and most connective tissues. Glycosaminoglycans, hyaluronic acid, dermatan sulfate, heparin sulfate are the hydrophilic components of the extracellular matrix that maintain tissue hydration and structural integrity. GHK-Cu upregulates the synthesis of both. This has been replicated in multiple cell culture systems and in animal wound models. It promotes angiogenesis, the formation of new blood vessels into healing tissue through upregulation of vascular endothelial growth factor, or VEGF. Blood vessel ingrowth is a rate-limiting step in wound healing. Without adequate vascularity, healing tissue is hypoxic and repair is impaired. GHK-Cu's angiogenic activity helps explain its consistent performance in wound healing models, and it activates anti-protease mechanisms. In wounds and in aging tissue, matrix metalloproteinases, enzymes that degrade extracellular matrix, are often overactive, breaking down newly synthesized collagen faster than it can be laid down. GHK-Cu has been shown to inhibit several MMPs, shifting the balance toward matrix deposition rather than degradation. The second major mechanism is antioxidant gene expression, and this is where the picture becomes more interesting from a longevity standpoint. GHK-Cu activates NRF2, the master transcription factor for the cellular antioxidant program. We discussed NRF2 in earlier episodes in this series. It controls the expression of Ham oxygenase 1, NQO1, superoxide dismutase, and glutathione-related enzymes. GHK-Cu's copper-dependent activation of superoxide dismutase is direct. It provides the copper cofactor that SOD requires. But beyond that direct mechanism, GHK-Cu appears to upregulate the broader NRF2-driven antioxidant program at the transcriptional level. Reduced oxidative stress burden, attenuated ROS-mediated cellular damage, and protection of mitochondrial function are the downstream consequences. In the context of aging, where oxidative stress is a well-characterized driver of cellular senescence and tissue dysfunction, this mechanism has obvious relevance. Third, anti-inflammatory signaling. GHK-Cu suppresses NF-kappa B, the primary transcription factor driving inflammatory gene expression. Its targets include TNF-alpha, IL-1 beta, IL-6, and a range of other pro-inflammatory cytokines that characterize the chronic low-grade inflammation, inflammaging, associated with biological aging. In cell culture models, GHK-Cu also suppresses TGF-beta 1 signaling through SMAD pathways, the same fibrotic pathway we discussed in the Clotho episode. Reduced TGF-beta 1 activity means reduced fibroblast to myofibroblast transition and attenuated tissue fibrosis. In the kidney, liver, and lung, the three organs where age-related fibrosis is most clinically significant, GHK-Cu's antifibrotic activity has been investigated in animal models with consistent results. Fourth, and this is the claim that requires the most qualification, is the large-scale gene expression modulation. A 2010 paper by Pickart and colleagues, published in the journal Genome Medicine, used bioinformatics analysis to identify human genes whose expression patterns are modulated by GHK. The analysis identified over 4,000 genes, upregulated genes clustered around tissue remodeling, antioxidant defense, neurotrophin signaling, and DNA repair. Downregulated genes included pro-inflammatory mediators, genes associated with cancer progression, and genes involved in matrix degradation. This is a striking finding. It is also one that requires significant contextual framing. Bioinformatic gene set analysis identifies correlations and patterns. In expression data, it does not establish that GHK-Cu is directly causing each of those gene expression changes through a specific receptor interaction. Many of those 4,000 genes are likely downstream consequences of a smaller number of primary mechanisms: RNRF2 activation, NF-kappa B suppression, TGF-beta modulation rather than direct GHK-Cu targets. The 4,000 gene figure is real and the analysis is peer-reviewed, but presenting it as GHK-Cu controls 4,000 genes overstates the mechanistic directness. It is more accurately described as GHK-Cu, through its primary mechanisms, appears to shift gene expression networks in directions broadly consistent with reduced aging-related pathology. One further mechanism worth noting is nerve growth factor upregulation. GHK-Cu has been shown in cell culture and some animal models to increase NGF expression and promote neuronal survival and axonal outgrowth. This is mechanistically coherent. Copper is a cofactor for several neurologically relevant enzymes, and it has attracted interest in the context of neuroprotection. The evidence base here is early, predominantly in vitro, and should be treated accordingly. This is where I need to draw a careful line. Because the gap between GHK-Cu's mechanistic story and its human clinical evidence is significant, and conflating them is the primary way this compound is misrepresented. The strongest human evidence for GHK-Cu is in topical wound healing. A number of clinical studies, mostly small, some randomized, have examined GHK-Cu in wound dressings, post-surgical wound care, and chronic wound management. The results have generally been positive: faster healing, improved cosmetic outcomes, reduced scar formation. A 1,994 study in the Journal of the American Academy of Dermatology examined GHK-Cu impregnated wound dressings in skin graft donor sites and found significantly faster reepithelialization compared to standard dressings. This is the kind of controlled clinical application to find wound type, objective healing endpoint, direct tissue delivery where the mechanistic picture translates most cleanly into a clinical outcome. Topical skin and hair applications represent the second area of human evidence. A number of studies have examined GHK-Cu in topical formulations for skin aging endpoints, skin laxity, fine lines, collagen density, and for hair growth in the context of androgenetic alopecia. The skin data, while limited by small sample sizes and variable methodology, is broadly consistent with the mechanistic expectation. Topically applied GHK-Cu appears to improve objective skin quality measures in several trials. The hair data is more preliminary, but similarly directional. These are not large Phase 3 trials. They are early phase and largely industry-supported studies. But they represent actual human evidence for actual outcomes in actual patients. And then we reach the limit of the human evidence base. For systemic or injectable GHK-Cu, the formulation being offered in longevity clinics and peptide practices, there are no completed randomized controlled trials with longevity-relevant endpoints. There is no Phase 2 trial data for injectable GHK-Cu in healthy aging adults. There is no established effective systemic dose. There is no pharmacokinetic profile established for injectable GHK-Cu in humans that would allow dosing to be rationally determined rather than empirically guessed. I want to be direct about what this means. It means that the systemic longevity application of GHK-Cu as it is currently being offered and used is based on extrapolation from topical wound healing evidence and preclinical mechanistic data. That extrapolation is mechanistically coherent. The biology makes sense, but mechanistic coherence is not clinical evidence, and the history of medicine is full of compounds with coherent mechanisms that failed to translate into meaningful clinical benefit or that produced unexpected harm at doses and in populations not studied in the preclinical work. This is not unique to GHK-Cu in this series. It is the standard situation for most longevity peptides. But it is worth stating clearly because GHK-Cu's preclinical story is compelling enough that it can create a false impression of clinical certainty that the evidence does not support. GHK-Cu does not hold regulatory approval as a pharmaceutical agent in the UK, EU, or United States for any systemic indication. Topical cosmetic formulations containing GHK-Cu are widely available and unregulated sold as skincare products without any requirement for clinical evidence of efficacy. This is the cosmetic regulatory framework. which is categorically different from the pharmaceutical regulatory framework. Injectable GHK-Cu, as used in peptide clinics and longevity practices, sits outside the licensed pharmaceutical framework in most jurisdictions. In the UK, a physician can prescribe it under their discretionary authority as an unlicensed medicinal product. This is legal. It is also a framework that places the burden of evidence assessment entirely on the prescribing physician rather than on a regulatory body that has reviewed the data. The asymmetry matters. It means that the quality of prescribing decisions varies enormously from clinicians who have engaged deeply with the literature and are making informed judgments to those who are prescribing based on anecdote and commercial pressure. The safety profile of GHK-Cu, insofar as it can be characterized, is reassuring in topical use. Systemic adverse effects have not been formally characterized in controlled human trials because those trials have not been conducted at scale. The preclinical safety data does not flag major concerns, but absence of a safety signal in preclinical work and small-scale clinical use is not the same as an established safety profile. It is a starting point. Here's where GHK-Cu sits in 2026. It is a naturally occurring, copper-binding tripeptide that declines dramatically with age, with plasma concentrations falling by over 60% between young adulthood and the 6th decade. It has a mechanistically coherent and unusually broad preclinical profile. Wound healing promotion through fibroblast activation, collagen synthesis, and angiogenesis. Antioxidant defense through NRF2 activation and SOD support. Anti-inflammatory activity through NF-kappa B and TGF-beta suppression, and large-scale gene expression modulation consistent with reduced aging-associated pathology. Its human evidence base is strongest for topical wound healing applications, where controlled clinical data exists. For systemic and injectable longevity applications, the evidence base is essentially preclinical. There are no completed randomized controlled trials of injectable GHK-Cu for systemic longevity endpoints. The optimal dose, frequency, route of administration, and patient population for any systemic application are not established by evidence. What we can say is this: GHK-Cu is among the most mechanistically plausible compounds in the longevity peptide space. The decline in its circulating levels with age is substantial and well characterized. The biology of what it does in tissue culture and animal models is coherent and consistent, and the leap from that foundation to clinical systemic use in healthy aging adults is a leap that the evidence has not yet caught up with. For those navigating this space, whether as patients or as clinicians, that distinction is the most important thing to hold on to. Mechanistic plausibility and clinical evidence are not the same thing. GHK-Cu has the former in abundance. The latter, for systemic longevity use, remains to be built. In the next episode, we'll be covering BPC-157, the most widely discussed peptide in performance and longevity clinic circles, and arguably the compound where the gap between the evidence people believe exists and the evidence that actually exists is most pronounced. We will do it the same way. Mechanism, evidence, honest gaps, no shortcuts. I'm Dr. Ethan Hausman-Markey. This is the science of ageless living.